Okay, so let's start. Good afternoon, everyone. I think because of Society for Neuroscience conference, most of the people there in the conference, so that's why we have less crowd. So, but however, we'll continue to discuss about um, growth cone. So, last class I talked about the axon grows at the tip of the growth cone. So I um, talked about how the basically how the growth cone looks like. So before going showing different regions of the growth cone, I just want to highlight some of these experiments are done to understand how growth happens at the tip of the growth cone. So this is this is experimental setup where people have done looked at the structure of growth cone. So here what they have done is they have placed a bead in the help of a micro needle, and they want to see how the growth cone respond to this bead. So what they have done here is, so here you can see that after some time, like after 4 minutes, some of this part of the growth cone, which I'm going to discuss, is extend towards the bead. So this is mainly when they did the staining, they found that this mainly consists of alpha and beta tubulin, and the peripheral part that you are seeing here consists of mostly the affectin, the filamentous actin. So if I go to the next slide, where the structure of growth cone has been discussed more detail in the snail. This is the snail growth cone. So here the growth cone consists of 3 different zones. So here you have the central zone where you have the microtubule consists of alpha and beta tubulin, which extends into the tail, like I've shown in the previous slide, extend to the tail like beads. And you have the transition zone in between the peripheral and central zone. You have the transition zone, and mostly the peripheral zone consists of the actin cytoskeleton. And from this peripheral zone, you have projection coming out from this peripheral zone. These are highly enriched with actin, forms the philopodia projections. And in between, and you have this kind of sheath-like in between the two philopodia. You have some structure that is called as lamillopodia. Actually, it forms like it attaches to the substrate, forms a sheath-like structure. These are flat-like structures present in between the philopodia. These are called as lamillopodia. So this is the structure of the growth cone. So and here it is diagrammatically shown like cartoonic representation of the growth cone. So here you can see that the central domain consists of the microtubule. So the microtubule, they forms the bundle, and it is present in the axon shaft. So this is the axon extending here, and it enters into this region, forms the central domain in the tip of the philopodia. So then you have in the peripheral region, you have this in the P domain, you have this actin cytoskeleton which polymerizes. The G actin polymerizes to form F actin bundle-like structure, and this is the one philopodia. In between the philopodia, you have the lamillopodia that I was talking about, like sheath kind of structure. So this is how the growth cone looks like. So we have 3 distinct regions: the peripheral region, this is the peripheral region, and this is the central region, and this is the transition zone. Okay. So in the transition zone, you have depolymerization of like the affectin depolymerizes, and in the P zone or the peripheral zone, it polymerizes to form the like bundle of affectin. So this zone, the transition zone, you have depolymerization of affectin, and this tip of this chilopodium, you have polymerization of the like G actin to form affectin. So this is how like 3 distinct compartment in a growth cone. Okay, and the central domain mostly enriched with microtubules, and peripheral region mostly enriched with actin filament, like F actin bundles. Okay. So here this slide I have already talked about before, but just I want to kind of show when the axon grows and how this this tubulin and actin cooperate each other to form the growth cone, active growth cone. So this is a axon which is actually growing, having the growing tip called as growth cone. So it is attaches to the substrate, and substrate. So you have certain receptor present in this in the growth cone called as integrin receptor that interacts with the substrate laminin receptor in the substrate. Then what happens is the granular actin, the G actin that I talked about before, they polymerize to form filamentous actin, the F actin, and they added to the tip of this growth cone. Now you need the membrane because you need actin, you need membrane so that the axon can grow. So the membrane is provided by the transported through this microtubule, stabilized microtubule, which is here. They transport this to the vesicles, to the tip of the growth cone, and because of the action of the actin, there is addition of a membrane and also growing of axon. So this is how the growth cone grows at the tip of the growth cone. So this mostly consists of the tip is highly enriched with actin. So here I'm showing different steps of growth of the growth cone. So here this is the axons, axon shaft. So it looks like a finger. You can see that these looks like finger, and this is the wrist. So they have designed the structure like that. So you have the axon here, axon shaft, then the wrist is here, then this is the C domain, the central domain, where you have the microtubules. The green are the microtubules, and these structure that you are seeing in black, these are the actin bundles. So what happened is in the axon shaft, the microtubules, the axon shaft, the microtubules are organized into parallel bundles with microtubule associated protein. So these structure that you are seeing in the blue, they are the microtubule associated protein MAP. So they in combination with or in working with they interact with the microtubules from the axon like microtubule bundle at the axon shaft. And then after that, the microtubules enters into the the C domain, the central domain. Then you have the T zone, the transition zone. So in the transition zone, you have severing of the actin cytoskeleton. You have depolymerization of the cytoskeleton from small bundle. You can see that these are the small broken actin bundle. Now, so what happens during growth of this growth cone? What happens is, this bundle that you are seeing, the microtubule bundle, they enters into the phylopodial, enters into the philopodia and forms bundle with the actin. Okay, so that you can see that this is extended to the tip, one of the philopodia, and then after that it remain extended. So and forms this bundle with actin bundle. So the microtubule forms along with interact with actin bundle to forms a bundle like kind of structure. So and here you have this structure that you are seeing here, the broken structure. They are broken. The actins are broken here in the transition zone because you have depolymerization in the transition zone, and then again the same thing that is extended to the phylopodium. So that is how the first you have in the axon shaft you have bundling of the microtubules along with the microtubule associated protein. It forms the bundle. Now this microtubule bundle enters into the central zone and enters into the tip of the philopodia and forms bundle with actin. The microtubule forms bundle with actin at the philopodia. Now, and when before forming bundle with actin, you have severing of this actin bundle in the transition zone because it enters into this region, this philopodia. So it sever this actin bundle in the transition zone. Then it forms bundle with the actin. So that is how the same thing shown here. Like you have the receptor. This is again the diagrammatical showing the same thing. How actin is actually polymerizing at the leading edge, and the growth of the axon is happening because of the interaction of microtubule and actin cytoskeleton, F actin cytoskeleton. So you have the receptor present in the in the cell. So those receptor actually interact with, those are called integrin receptor. They interact with the substrate. Now they give signals. So they give signals, and those signals actually regulate the polymerization of a G actin. That regulate the cytoskeleton to polymerize the G actin and the extension of extensor or microtubule at the tip of this this growing growth cone. So that's how the axon growth takes place. Okay. So here the more detail showing how tip of the philopodia. So here this is one philopodia is showing here. So here in the transition zone you have depolymerization of the actin cytoskeleton, because you have the depolymerizes. You have entry of the microtubule, and also you have depolymerization of the this actin cytoskeleton from the G actin, the granular actin, and the granular actin transported to the tip of the philopodia. And after transported to the tip of the philopodia, they again come back. They came back to this direction. There is a retrograde flow of the you have the G actin. So first at the transition zone they depolymerize. So this is the transition zone, and they again transported. And this type of transport is called as actin treadmilling. Again, so it transported to the tip of the philopodia. So it enters when it reaches to the tip of the philopodia. Again, it comes back to the central, this region called as this type of transport is called as a retrograde F actin flow, and that's how you have addition of the actin and growing of the philopodial tip. Okay, and you have protrusion of philopodia. So this type of flow is called as F actin retrograde flow. Okay, so this is showing same thing here. So this actually flows to this region. This is called and after flow retrograde flow, they when they attaches to the substrate. So what happens is first the growth cone attaches to the substrate, and when they attaches to the substrate, because it cannot grow without attaching to the substrate. Once they attaches to the substrate, you have depolymerization of the actin at the transition zone. Then you have transport of this G actin to the tip of the philopodia, and then again they come to this region forming the actin clutch. So this type of this is how the growth cone happens. The growth happens at the tip of the growth cone. Formation of actin clutch, because you have transport of this G actin and formation of the F actin in this region. Okay, so F actin continues to polymerize at the leading edge, and you have the growth of the growth cone happens because of the continuous polymerization at the tip of the philopodia. So this is a movie showing how the growth cone happens. You can I'll play it again. So you can see that this is growing again, coming back. This coming back is the retrograde flow of the actin. So if I can play it again, you can able to know. So this is the tip is growing, but it is also retracting. Retracting means the actin is actin is coming back, forming actin clutch. Okay, so that's why you have growth, and also you are seeing such type of movement. So here this is the structure showing this philopodia is not engaged. The growth cone of this philopodia is not engaged because you don't have formation of don't have formation of the bundle with actin. The microtubule is not there, so you don't have because you need the microtubule as well as association of microtubule as well as actin, the filamentous actin, for the growth of the philopodium or growth of the growth cone. So here this growth cone is not actively engaged with growth. So but when this philopodia get some substances called as chemoattractant, because you need external cues for the growth of the growth cone. So the growth cone can grow towards the chemoattractant, towards the substance which can so that type of movement is called as axon is pathfinding. Axon is searching where it can bind, where it can go. So when it finds this chemoattractant, you have the growth this microtubule which is present in the central zone. They extend to the philopodium, forms bundle with actin. So this is the actively growing growth cone, the actively growing philopodia. Okay, so that is how the growth cone the growth happens at the growth cone. So here showing that same thing like cartoonically. So here you can see that so here the microtubule actually it's extended to the philopodia and making bundle with actin. Here so this is so this type of these are the peripheral microtubules, but microtubule can act as so basically what function of microtubule here is the microtubule enters into the philopodia and serving as a scaffolding protein. Scaffolding protein means it is supporting the actin so that the philopodia can grow easily. Philopodia can grow easily. So it acting as a scaffolding protein, and also it is scaffolding protein for guidance Q signaling. So it is helping the philopodia. It is helping the actin, F actin, to grow at the tip of the philopodia. So it is acting as a scaffolding protein, supporting protein. Okay. So and this type of structure you will find in the actively growing philopodia. Okay, so here this cell is labeled with microtubule binding microtubule and binding protein EB3 and actin. So you can see that the green is the microtubules and the red is the actin. So you can see that it is forming bundle. It is forming bundle with actin in this philopodia. So if I play it again, and you can see that in this philopodia it is forming bundle. So this is an actively growing philopodia. Okay. So again, if I summarize everything what I have said for the growth of the philopodia or growth of the growth cone. So here in the first stage, what happens is when the growth cone encounter a substrate or a chemoattractant. So chemoattractant, what happens is you have so this is the axon. You have in the central zone you have the microtubules. The microtubule try to extend its microtubule from the central zone to the peripheral zone, so P domain, the P zone. So it starts extending its microtubules. Then you can see that the microtubule projects. Here the domain is the domain of the microtubule is little is smaller here. It's kind of rounded here, but it becomes narrower here, and it extends into the philopodia, forms bundle with actin. And this stage is called so this is the third stage. The second stage is the protrusion. So what happens is so after it encounters a substrate, it extends its microtubule to the philopodia, and also it forms a bundle. Then this stage is called as a protrusion stage. It protrudes means it extends its microtubule to the tip of the philopodia and forms a bundle with actin. Now after forming the bundle with actin, you can see that how this the microtubule, it was wider here, but it becomes narrower here. And the C domains, the central domain where the microtubule is present here, it extends, it moves forward, and so that the microtubule can enter into the actin bundle. And after formation of the actin bundle, then you have efficient generation of philopodia. And this structure that you are seeing here, the axon shaft here, it becomes narrower, much narrower, because of the tension generated because of you have growth of these microtubules. It kind of pull out. So that's why you have the narrower of this axon. So this is how you have growth of the philopodia, or the growth of the growth happens at the growth cone, interaction with actin and microtubules. So any question before going to the next session? Any questions? Okay. So so here so this is I showed you molecularly how growth happens at the growth cone, and how the actin and microtubule interacts to form actin bundles at a different region of the growth cone. So here this is the diagram showing the part of the brain in the thalamus called as lateral geniculate nucleus. So here showing that how the growing axon, it is not only the interaction of actin and microtubules. There are a lot of thing happens in between. After that, before when the axon grows in the brain and finds its target, a lot of thing happens. It's not only it is not only the interaction of actin or microtubules. There are a lot of thing happens. So here it is showing in this region of the brain, the lateral geniculate nucleus is a part of the thalamus, which is required for the visual processing. It is showing that how growth axon targeted to different region of this brain. So there are a lot of things happens in between. So we'll go like molecularly how this thing happens, how the axon is guided to different part of the brain, how it is grows in different target to the different part of the brain. So here showing different steps of the axon guidance or the axon growth. So what happens is, so here this is a neuron, and here this is the cell body, and this is the axon. So first first step of the axon guidance is the interaction with the extracellular matrix. So ECM, so extracellular matrix adhesion. First, it attaches to the extracellular matrix, binds to the extracellular matrix. Then, the second step is it binds to the surface of other cells, like it is called a cell surface adhesion. So this is another neuron which has a surface. Then it extends its axon and binds to the surface of another neuron. So second step is cell surface adhesion. The third step is fasciculation. It forms bundle with the existing neuron. So this is the pioneering neuron, the existing neuron. It has its own axon. Then first what it does is, it extends its axon, binds to the extracellular matrix, which is here. Then it attaches to the surface of other axon, so pioneering axon. So this is and forms a bundle. So this is called as fasciculation stage. Then after fasciculation, then depending upon the signal, if it is a attractant or chemo repellent or chemorepellent, what happens is they send their axon. If it is a positive signal, it grows in that direction. So first step is extracellular matrix adhesion, then cell adhesion, then fasciculation or forming the bundle, then chemoattraction or chemorepulsion, depending upon the signal. So here is a chemoattraction signal. So it can go to this region because there are signaling molecules which can attract, which can help in the growth of the axon terminal. So this is the chemoattraction, or it can repel this signal and bind to another cell. So this is called a chemorepulsion. So depending upon the signal, it goes in that direction. Then another step is a contact inhibition. Sometimes what happens is, the axon is growing when it finds an inhibitory substance, it stops growing. It is called as contact inhibition. So these are different steps of axon guidance. It is not only the polymerization of F-actin or microtubules. So a lot of things, a lot of steps in between, like after that. So by the combination of actin, tubulin, or by combinatorial, axon of this, you have growth of axon happens at the tip of the growth cone. So this is diagrammatically represented here. So here this is showing the chemoattractant. So it can attract to the positive substance, the positive signaling molecule. So this is then there are two types of so they are categorized into 4 different types, which is contact mediated repulsion, contact mediated attraction, diffusible chemorepellents, or diffusible chemoattractants. So what happens here? This is the one axon. So it is this is another axon. It's forming kind of bundle. So it attaches to this axon. So this is called as contact mediated attraction. So here it is not attracted. This axon is not attracted to this axon because this is negatively some kind of signaling molecule is secreting, which is inhibitory kind of type. So it is not attracted to this. This is called as contact mediated repulsion. And sometimes diffusible attractant are there, diffusible repellent are there. This is the diffusible attractant is growing to that direction. This is diffusible retractant. So showing different molecules for the axon guidance. It is it's very interesting. So we will talk about some of these molecules, diffusible molecules, or or we will talk about some contact mediated attraction or contact mediated repulsion. Some of the examples. So I'm going to talk about. So this is this slide shows how Axon, extracellular matrix molecule helping growth of the axon. So how it is guiding the axon in a proper direction. So this is one cell, this is another cell, and this is the extracellular matrix, the ECM, and you have a specific receptor present in either of the cells. So this is the integrin receptor, and you have the in other cells you have the another integrin receptor. So this is one neuron or axon, you can say it's another axon. So what happens is, so the ECM interact with this the extracellular matrix interacts with this integrin molecules and initiates an intracellular signaling cascade, which can regulate the underlying cytoskeleton that I talked about, actin or tubulin. And it can regulate the dynamics of actin and tubulin to regulate the growth of the growth cone. So they are both attractive and repulsive. There are some of the molecule or these molecules which are present here, the extracellular matrix. They can be act as attractive molecule or repulsive molecules, repulsive extracellular matrix molecules. So depending upon the signal, you have either growth of the axon or you have inhibition of the growth of the axon. So this is the example of extracellular matrix molecule, how it is helping axon to grow. So now we'll talk about another cell to cell, that is cell to ECM molecules. So this is cell to cell. So there can be interaction between cell to cell. So in one cell, you have cell adhesion molecules. So this is called as this is one of the cell adhesion molecule present in this cell. This is called as L1. And another cell, you have another cell adhesion molecule called as neuronal cell adhesion molecule, and these two axons, they are in kind of coming together, and so when they come contact together, so they interact and again initiates intracellular signaling cascade, which can regulate the underlying actin and tubulin cytoskeleton. So then you have addition of the G actin, polymerization of the F actin. Then you have growth of the growth cone. So another cell-to-cell contact molecule called as cadherin. So this is one axon, this is another axon, same thing. So you have when this cadherin interacts with, it binds with, so in these cells, you have the cadherin which binds with calcium and become activated. So interact with cadherin of another cells. Then you have activation of beta catenin and alpha catenin. So and cadherins are mostly permissive signal, so they allows polymerization of actin. So they helps in polymerization of actin. That's why you have extension of philopodia, and this is just to show that how cadherin helping polymerization of the actin and growth of the growth cone. Very simple. So these are a lot of the kind of signaling molecules showing that how they are helping or dynamics, or they're regulating dynamics of actin and tubulin for the growth at the tip of the growth cone. So, and some of these repulsive signal also present in cell to cell contact models. So here this axon, this is one axon, this is another axon. So they are interacting with each other through the specific type of receptor. So in this axon, you have the semaphorin, and in this axon, you have the neutropilin, okay, or plexin. So plexin, we know that from previous lectures, plexin interacts with semaphorin. So when you have, and these are the semaphorin are generally repulsive signal. So one cell is sending the repulsive signal. So this will not definitely attracted to that axon. It will stay away from that axon. So what happens is when the semaphorin interact with neutropilin or plexin, it activates the downstream signal, which is Rho and Rho gap. When Rho gap is activated, it depolymerizes the actin. This is a you have dissolution of the you don't have the formation of enough F actin, so you require F actin polymerization at the tip of the philopodia for the growth of the growth cone. So here the depolymerization happens. That means you don't have the growth cone doesn't grow, it stops. So because this is a repulsive signal, the semaphorin. So and because you have activation of Rho gap because of the signaling through the semaphorins. So then other type of signal which is afferent signals. So afferent, this cell secretes afferent signal. You have afferent EPH. The afferent ligands binds to this afferent receptor. Okay, so this is showing that the afferent again is a repulsive signal. So it when it interacts with other receptor present in other cell like other axon, it again activate the Rho gap kind of signaling, which is depolymerizes the actin. So and then you have the tip of the philopodia cannot grow. It stops its growth. So because the afferent or the semaphorin, they are the repulsive signals. So here just showing that cell to cell contact it is highly essential for the axon guidance. Okay, so the Rho gap mostly depolymerizes this F-actin to G-actin. Then other other diffusible. So I showed you some of the ECM molecules that is acting as a chemoattractant or chemorepellent. Then the cell to cell adhesion molecules acting as a chemoattractant or chemorepellent, and these are the diffusible molecules. Some of these diffusible molecules can also help for the guidance of the axon. Okay, so this is one axon, this is another axon. So this axon secreting one of the diffusible molecule called as natrin. So and also it secretes another repulsive molecules called as so it's showing that two distinct kind of signaling molecule, diffusible molecules. So natrin act as a chemoattractant. So here natrin binds to this DCC, deleted in colorectal cancer receptor. So DCC, then it polymerizes polymerizes to the actin and growth of the philopodia. But however, slit secreted from these cells, it interact with the robo of another cells, another axon, the robo receptor, and it activates the Rho gap. So here the Rho gap has dual rule. Depending upon the ligand, it will polymerize. So it is not only always it will depolymerize. It sometimes here you can see it polymerizes when it finds these natrin ligands, but when it finds slit ligand, it depolymerizes the Rho gap. So it is a repulsive signaling molecules. So depending upon the need, the cell will activate either of the signaling cascade to polymerize or depolymerize the actin molecules at the tip of the philopodia. So the Rho gap. So here this is summary showing that different kind of attractant or repellent and kind of helping the philopodia to grow. So here showing that how natrin act as attractive signals, showing this side you can see that there is a growth of the philopodia, and this side you don't have the growth of the philopodia because these are the repulsive signals such as semaphorins, slit, and all these things. And when you have generation of these signals, semaphorin 3A or slit, you have loss of the actin filament, loss of the dynamics of the microtubule because it activates Rho gap signaling, which basically depolymerizes the actin cytoskeleton, except this case, natrin case. So it polymerizes because it is actually attractant for the growth cone. So these are the summary, and this is the model for the growth cone guidance, and showing the same thing here, like how the growth cone tips is organized. So here information of the axon, information of the microtubule bundle, the microtubules. These are the repulsive signal that I am showing here because you have activation of the Rho gap signaling, and you have depolymerization of the cytoskeleton, the actin cytoskeleton or tubulin cytoskeleton. That's why you have retraction of this philopodia, and here you have a polymerization of this active polymerization of the cytoskeleton. That's why you have growth of the lamellopodia or philopodia. So showing same thing, the slides. So here I'll talk about some of these, more attractive cues or repulsive cues belongs to, and I'll just want to highlight how road GTPases couple guidance cues to the active cytoskeleton. So here, when it binds to the attractive cues, so this axon, what happens is activate these sets of GTPases, small GTPases, which can catalyze GTP to GDP. So the CDC42 RAC1, these are the GTPases. So when you have attractive cues, you have growth of the growth cone and growth of the axon. So because you have activation of these small GTPases, and you have formation of filopodia, lamellopodia, and that's why you have formation of this growth, the growth of the growth cone. But when you have repulsive cues binds to these, you have activation of row gap, or row A signalings that I was talked about before. When you have activation of row A signaling, it depolymerizes the actin cytoskeleton, and you have retraction of filopodia or lamellopodia. Okay, because you have depolymerization of this, and the polymerization is a highly essential for formation of filopodia. Since you have depolymerization of this, microtubules or actin cytoskeleton, you have retraction of this filopodia. So again, showing that the same thing here, and this is highly complex. So here the extracellular ligands binds to the receptor, and it generates sets of adapter, and these adapter act on the small GTPases, either CDC42 RAC1 GTPases, which act as attractant, or which initiates the polymerization of the G actin to form F actin. So it forms the filopodia, or it activates the row A, where you have depolymerization of the filopodia and you have collapse of the growth. Okay, so remember that row A is a kind of inhibitor for formation of filopodia. It depolymerizes the actin and tubulin, row A. Okay, row A, and these are the again. These GTPases helps in formation of F actin from G actin, granular actin. So they act as a positive regulator of filopodia generation. This CDC42 and RAC1 is a positive regulator of filopodia generation, and Rho A is a negative regulator of filopodia generation. Okay, formation. So here showing that different steps of filopodia generation by activated Rho A and interaction with actin and tubulins. So so this is what happens in the first step. So you have this chemoattractant or that binds to the receptor here. So this is the second step. What happens is when you have so initially what happens, you have less Rho A signals. So you can see that you have very less, and you have more RAC or active molecules. That's why you have filopodia here. So when it binds to this, what happens is the signal binds to this. You have inactive. You have RAC1 become inactive, and this become active. Which one? The Rho 1 become active. When Rho A become active, it depolymerizes. After 6 to 12 minutes, it depolymerizes this. Okay, so you can see that you have loss of this filopodia. Then after some time, you have this RAC1, which was inactive, become active. So and when RAC1 become active, it forms flat-like membrane. These are called as lamellopodia. So what happens is first you have activation of RAC1, so that you can see that there is a flat-like structure formed here. This is because of activation of RAC1. It forms lamellopodia. Then filopodia is formed because of polymerization of the actin. So when the attractant binds to the receptor, you have activation of Rho A, or activation of Rho A, and Rho A depolymerizes the actin. So you have dissolution of this actin, and you can see that you have loss of this filopodia. Now, after sometimes the RAC1 become active, so you have formation of the sheath-like structure that is called as lamellopodia. So this is the vesicles and the membrane that you are seeing, the resolution of the membrane happens because of the formation of endocytic vesicles, and that is happen because of the activation of Rho A. Okay, so the membrane will be engulfed. The filopodia will be dissolved and forms membrane here, and this will come to this to this tip of this growing growth cone. So it will add the membrane and forms a flat-like structure. So this is called as lamellopodia. So the RAC1 become activated at the final stage because it forms sheath-like structure, the lamellopodia, and this dissolution of this actin that happens because of activation of RhoA. Okay, so this is how the growth cone, the dynamics of cytoskeleton regulated by the interaction of RAC1 or RhoA. Okay, so here this is the experiment shown in the lab, how RAC regulates collapse of the growth cone. So here here this vehicle, the DMSO treated sample, you can see that you have the nice growth of the growth cone, you have the filopodia coming out, and here the RAC1 is inhibited. The RAC is inhibited. So there are some drugs which you can use, which can inhibit the RAC. So here the RAC1 is inhibited so that you don't have formation of sheath-like structure, because RAC1 forms lamellopodia, and here you can still see the filopodia-like projection. But when they add the afferent signal, which is a repellent signal, so you can see that. So here the vehicle is there, so you have less collapse of the growth cone. You have RAC1 inhibitor. So because RAC1, what it does is it forms lamellopodia. So it forms lamellopodia because if you look at this stage, the collapse of this growth cone requires RAC1, because you can see that the RAC1 molecule become activated at the end stage of this development. So you can see that more RAC1. So here when they inhibited the RAC1, you have less collapse of the growth cone. That's why you can see that you have less collapse of the growth cone. But when they add the afferent A2 signal in the vehicle, you can see that there is more collapse of the growth cone. You can see that the growth cone collapse because you have the afferent is a repellent. It activates RhoA signaling. It depolymerizes the actin. That's why you have collapse of the growth cone. You have more collapse of the growth cone. Okay, but when they increase the concentration of RAC1, like 0.5 molar, 0.75 molar, or 1 molar or 2 molar, when they increase the concentration of the RAC1 inhibitor, you can see that the collapse of the growth cone goes down, which is shown here, even in the presence of afferent A signal, because afferent A collapse growth cone because it activates RhoA. It depolymerizes the actin. That's why you have collapse of the growth cone because of afferent A2 signal, afferent A2 treatment. But when you use this RAC1 inhibitor, okay, RAC1 will not be activated, so that the collapse will not happen. So that's why you have less collapse. Okay, this is just showing that how RAC and afferent regulate growth cone development. So this is the summary slide showing showing how this different GTPases regulate growth of the growth cone, or it inhibits the growth cone. So here showing that the RAC, when you have formation of RhoA, it can signal to the ROC can signal to the limb kinase. Limb kinase inhibits coffulin. When the inhibition of coffulin happens, then you have the growth inhibition, showing that how Rho actually regulates this pathway for inhibition of the growth cone. And here showing that how RAC is regulating polymerization of the how is RAC is regulating the actin dynamics. So the interaction of RAC and Rho regulate actin dynamics and growth of the growth cone. Here's showing that how the cell uses different receptor and different adapter to recruit different GTPases. So here afferent A recruits afferent A4 adapter, afferent B recruits afferent B2 adapter. So afferent A regulate this signaling pathway, Rho pathway. It can regulate ROC, which can inhibits the growth cone. So showing kind of complex things, but so I am not going to put question on these slides because this is something signaling network, and I think this something difficult to remember. So I am not going to put a question on this slide. So here showing how different receptor utilizes different adapter recruit RORAC and all these things. So here the slit robo interaction. So it recruits this set of adapter and we act as a repulsive signals. So then natrine DCC recruit this set of adapter, like it regulate polymerization of the effectin. It act as attraction, attracting molecules or attract signals. So the same thing can different receptor can utilize different can generate different adapter to regulate either growth of the growth cone or inhibition of the growth. So the most important thing that I want to show, when the growth happens at the tip of the growth cone, there if you look at molecularly, so they have seen that the calcium actually increases at the tip of the philopodia during the growth of the growth cone. So here this is typical structure, the part of the growth cone labeled with two different dye. So this is a fluoro-4 dye which can which can give signal, so fluorescence at the green in the green range. So here the fluora red, it can give signal in the red range. So both are the calcium indicator. So, and this is just to normalize this this GFP signal. They have used this one fluora red. So you can see that after 250 milliseconds, after this is 0 millisecond, so after 125 millisecond, when there is some attractant binds to this growth cone, you have after 125 millisecond, you can see that. So this is the range indicator showing the signal. So blue means you have less signal, red means you have more signal, kind of 3 is intermediate, like kind of a yellow red mixture. So you can see that after 25 millisecond, you have the philopodial tip is highly labeled with fluora-4. So that means fluora bind to the fluoro-4 bind to this calcium. So that's why you have the signal is coming here. So then they found that attractive cues actually increases when they incubate this growing philopodial tip or the growing growth cone with attractant. They found that there is a higher in concentration of calcium accumulated at the tip of the philopodia. Okay, so you can see that the philopodial tip has a higher concentration of calciums. So what they have done is they have measured the change in calcium signals. So first, without putting the attractant, they measure what is the signal for the calcium. After putting the attractant, after 250 milliseconds, they measure the like signal for the calcium. So they subtracted that. They found this change in calcium fluorescence, and they plotted here. You can see that as the time grows, you have more calcium at the tip of the philopodia, and here the same thing plotted here, like in the below panel. So here they have used a inhibitor for they eventually found that the calcium entered to the tip of the philopodia through a cation channel called as TRP channels. So when they block this TRP channel, is a cation channel with this inhibitor SKF 96365 inhibitor is a block for the TRP channel, and the calcium enters through the TRP channel. So when they incubate that with this signal, so natrine is a positive attractant. So here in the control, they incubate this philopodia or this growth cone with natrine-1. You can see that the calcium signal increases as the time progresses, like 0 minute, 1 minute, 2 minute, 5 minute, 10 minute. You can see that the calcium signal increases. That means the natrine-1 causing to increase the calcium signal at the tip of the philopodia. But when they use this SKF, which is a blocker for TRP channel, they found that you don't see such increase in calcium concentration in the tip of the philopodia because it blocks this channel, TRP channel. So that's why you have decrease in fluorescence, change in fluorescence even if you have treated with natrine-1. Okay, because you have blocked this channel, TRP channel here. So the conclusion is that you have local increase in calcium concentration at the tip of the philopodia when this philopodia finds an attractant molecule like natrine-1. Okay, that's what is shown here. And when they look to more detail into this, they found that the increased calcium concentration at the tip of the filopodia contribute to the growth of the growth cone guidance. It guides the growth cone. So they found that the overall filopodia length or overall filopodia number is not affected. So here you can see, so they have used two attractant molecule. One is netrin 1, another is BDNF. So here in the control, you can see that they measure the neurite growth in the control, or the control treated with netrin 1 plus SKF, the inhibitor. They found that the overall growth is not affected. The neurite growth is not affected. Or with BDNFs, in all the conditions, they found that the neurite growth is not affected. It is only the the neurite is turning their angle depending upon the calcium signal. So here you have this control. So netrin 1, you can see that they are growing nicely, they are projecting to different regions. But when they treated with SKF, you have loss of calcium. You don't have local increase in calcium concentration because you block with TRP channel with this drug. Then you can see that the overall growth is fine, but the turning angle is affected. Here they are turning nicely, here they are not turning. That means they are not projecting to the proper project, they are supposed to project. So here the neurite growth is not affected by SKF, but the turning towards attractant is prevented. That means the local increase in calcium concentration is required for the axon guidance. Okay, and that is happens because you have activation of the TRP channel, which allows to enter calcium to the tip of the filopodia. Then you have turning of this axon like the filopodia to different region depending upon the signal, where the signal is coming. So that's how the axon guidance happens with combination of this calcium signal with TRP channels and along with the attractive molecules. So that is kind of discussed. So here it is not only the attractive molecules, it is the when the axon growth happens, there is it generates a force called as axon mechanical tension that can also helps in growth of the axon. So here this is experiment is kind of here describing how mechanical tension causes axon growth. So here this is one cell. So here this is a bead in the in the microelectrode. So when this axon attaches to this bead, so it becomes like kind of it helps in kind of growing that axon towards the direction. So it will be shown morely mechanistically here. What happens here? So you have the cytoskeleton, the microtubule present in this axon. So this is shown here. And there are some mobile microtubule segment are also present. So when the axon attaches to a substrate, what happens is you have removal of this axon microtubule bundle in this region, and you have this mobile segment, they move to this region, and this region is become thinning because you don't have this supporting cytoskeleton. Okay, so axon cannot grow cannot get a proper structure. So when it becomes thin, it can actually pull when you have the tension can actually act on this, so it can actually pull and it can grow easily. So the mechanical tension actually helping the axon to grow, and you have local thinning because you have removal of the microtubule, you have the mobile segment, microtubule segment, and moves here, then you have tension created, then you have growth of the axons. So it is not only the calcium signal, the mechanical tension also causes axon growth. So these are some of the key points showing that actin polymerization required for the axon growth. Microtubule extended to the filopodia forms scaffold and helps in axon growth. How different small GTPases go through these key points what I've discussed so far. So is required for the axon growth, and also the mechanical tension is required for the axon growth. So here the showing same thing like how axon guidance happens, kind of summary slide. You have diffusible chemoattractant, diffusible chemorepellent, or contact mediated repulsion because of a negative signal, or contact mediated attraction, and the axon, they grow together forming a they stuck kind of forming a bundle called as fascicular like growth of axon. So that is kind of I'm highlighted here these things. So if I summarize all the axon guidance mechanisms, so guidance can be innate guidance. So innate guidance means it's already there. So and the axon knows that I will go to this place. So that is predetermined. The fate is determined. That is called innate guidance, or through the extracellular matrix cues, through the extracellular matrix, or sometimes the one neuron can also guide to the other neuron to to target to the different region. This is through the pioneering neuron that I talked about before. So this is example showing how pioneering neuron actually helping for the growth of the axon. So this is a grasshopper embryo showing how one cell guides the other cell to reach to its target. So the target is abdominal ganglion. So these cells called as TI1 cells. Thus, they project to the abdominal ganglia. And these cells that you are seeing here, the guide post cell, that guides these TI1 cells to reach to the abdominal ganglia. So when this is the normal scenario, you can see that these cells, the guide post cells, the guiding this axon to the central nervous system. But when you have loss of these cells, the guide cells here, the axon cannot grow the axon cannot go to the central nervous system. Rather, you can see that it is growing inward. So this is the showing that how different mechanism of axon growth. It is through the chemoattractant or chemorepellent, or through mechanical tension, or through polymerization of the F-actin, or the polymerization of the microtubule, or through the pioneering neurons. The guide post cells can also guide one axon to to their targets. So this is the final slide showing how this thing happens, how one axon can guide to the other axon to reach to the target. So this is the pioneering axon, the pioneering neurons. So here this is the extracellular matrix. So you have these molecules called as the laminin molecules, and in the pioneering axons, you have these molecules called as integrin. So integrin attaches to the laminin, and then what happens is you have the generation of the signals, which can regulate the cytoskeleton, the actin tubulin cytoskeleton, and you have the growth of this filopodial, like the growth cone. Then when another axon interact with this axon, so this axon is helping this axon to reach to the target. So that's why it is called as a pioneering axon, pioneer axon. Okay. So you have cell surface molecules present in each of these axons called as cell adhesion molecules. It can be cadherins and other molecules. So they interact with this pioneering axon, and because of the interaction, you have the initiation of the downstream signal, and which can actually polymerizes the G-actin or F-actin, and you have the addition of this membrane and growth of the axon along the pioneering axon. So this is showing the same thing, kind of highlighted different types of mechanism for the axon guidance, and so it's highlighting chemorepulsion, chemoattractant. So this is all about for the axon growth and guidance. So if you have any question, we can discuss more about this. So showing that different mechanism for the axon growth and guidance. So it's not only the actin tubulin, it is also the mechanical tension, or by the help of the pioneering axon. Okay.